Hi, I'm Henry, and I'm going to present one of my PhD projects on proteomics data denoising and imputation using or self-supervised deep learning. My supervisors on the project are Anna-Laura Bach-Nielsen and Simon Rasmussen, and we are all three from the Novo Nordisk Foundation Center for Protein Research in Copenhagen, Denmark. The poster presents the dataset we're using in this project, the data views which can be generated from this dataset, a brief section on the motivation, the models we use currently, as well as evaluation strategies and envisioned applications. I begin this video with a brief description of the dataset. Each dot in this principal component plot here represents a sample, and these samples are constituted of peptides, and more specifically the 1,000 most abundant peptides shared by all these datasets, and they are color-coded by the date of measurement. We can see that they cluster by date in their first two principal components, and we aim to basically make these samples comparable and to impute missing peptides in them. From this 1,000 most abundant peptide dataset, one can generate different data views. Here, the values you see are log transformed intensities, normally log 2 or log 10. And in a long data format, each sample is associated with a specific peptide, and an intensity measure is recorded alongside. Some samples will have not available as a value for not measured peptides, and the long format can be translated in a different data view into basically the wide format, where the input row, so each row is an input, where the input row basically views the correlation between the peptides, or an input row, the peptide basically views the correlation between the samples. From this dataset, a brief jump motivation. In downstream applications, some researchers I ask, use a shifted normal distribution now for the imputation task. So if they have aggregated protein intensities here represented by the blue distribution between different samples, they basically shift for a protein the normal distribution to the negative part, the low abundant part, reduce the variance, and draw for imputing missing proteins samples from this green distribution. And we are basically working on this task to improve this kind of imputation technique alongside with hopefully denoising. The two envisioned applications are denoising and imputation. Denoising focuses on making samples comparable, so also changing measured peptides, whereas imputation only focuses on the replacement of not measured peptides, or imputing the not measured peptides, and some models are inherently capable of both. We're discussing currently if models should be always transferable to new data, or if they can be retrained on each data set, and how this affects the composition of the training, validation, and testing data set. And in general, we would be happy for discussions on what does it mean for differential analysis and downstream prediction task, and how could this be assessed. The models. In the Autoencoders setup, we would use the wide format, currently the peptide correlation format, where each input is the peptides measured for a sample. And this basically sample peptides are encoded into a hidden representation, from they are decoded, and the reconstruction should be a denoised or imputed version of the input. The two architectures considered are denoising autoencoders, which have a deterministic hidden representation, and variational autoencoders, where the hidden representation is stochastic, so each decoding step will yield slightly different reconstructions. Setup is the collaborative filtering, where each peptide and each sample are assigned an encoding, and the combination of both in a decoding step, which can be a neural network or just a scalar product of both, yields the reconstruction of the log intensities. So this setup would use the long data format. The inherent limitation is that probably one needs to create a sample embedding for every new data set, except one has another model for predicting sample encodings. Which leads to how do we compare these different models in their quality. If we only focus on the reconstruction error, currently the collaborative filtering models in our setups yield best results in comparison to denoising or variational encoder. If we focus on the basically reconstruction, we also might focus on the biases inherent in the data. So for a denoising setup, the best reconstruction might not always be the desired outcome. That said, there are many open questions to answer, and we would be happy to discuss any aspect of our current project. Thanks for listening.